Okay, this is chapter 18, Adaptive Immune System, Your Specific Host Defenses. This is just a nice picture of how a polio epidemic really changed America in terms of vaccinations. Polio was a very common disease in the 1950s, and the virus, the polio virus, could infect the nervous system and cause paralysis. So you can see all these children or people in these iron lungs because they needed a pressure system to help them breathe. They were paralyzed and so unable to breathe for themselves, which is really scary. So vaccinations are part of this chapter. We'll learn about how they work. In the immune response, your specific immune response, there's two phases to it. So the initial exposure to any pathogen, you're going to get a very slow rise in your primary immune response. And this is basically the antibody concentration in your body. Antibodies are made by your B cells, which are part of your specific immunity. And then they wane over time. And then what's created and what causes a very quicker, right? So you can see the upward line here to increase your concentration of antibody is much faster than this one. It also is almost triple the amount of antibodies total in the second immune response, because what the primary immune response did is make memory cells. And so your specific immunity can make basically these cells that remember the initial pathogen that came in. And so the next time you get exposed to that same pathogen, those memory cells react, and the memory cells create a much stronger and faster reaction. And this is just can become natural. You can get naturally infected by the same maybe cold virus twice. Or it could be, this is how we use vaccines, because vaccines cause your body to make a primary immune response. And then your secondary exposure would be the real guy, right? The real pathogen in the world. And so your body is going to make antibodies much, much faster and much, much more to protect you against that actual infection. So let's talk about antigens. Antigens are any molecule that can provoke a specific immune response. And in fact, antigen word came from antibody generating. So they are going to alert your specific immune system to act against it. Antigens don't have to be foreign. They can be your cells. So the way your immune system can learn your cells from other cells is that they can recognize antigens on the surface of your body, of your cells, and leave them alone. Okay. However, if you think about blood types, if you gave someone the wrong blood type, right, then that person would destroy your tissues, your blood tissues. And so antigens can be, normal parts of your body, but if it's foreign to someone else, then they're going to receive, the immune system is going to perceive that as foreign. So really quickly, antigens can be anything. They can be part of microbes, obviously. Those are called extracellular antigens or exogenous antigens. They come from outside the cell, so they can be parts of the wall. Maybe this is peptidoglycan or LPS. They can be part of the flagella. Remember, there's a protein called flagellin. They can be spikes or proteins of a virus. They can also be toxins, so anything that's arising from outside of your own body is called an exogenous antigen. Then there's endogenous antigens, which we have made inside of our cells. So a classic example is a viral infection. The viruses come into our cells process pieces of that virus, and our cells are able to put pieces of that virus up onto the surface of the cells, basically alerting our immune system that we've been infected by virus. And those, because the antigen came from inside the cell, it's an intracellular, from an intracellular virus, it's called an endogenous antigen. And then there's these autoantigens. So this is going to be what I was talking about, our normal cell antigens. our immune system can detect whether these surface markers of our cells are normal or abnormal. And if they are abnormal, then our specific immune cells can destroy that cell. So an abnormal example of an abnormal cell would be a cancer cell, where when cells undergo the transition to cancer, these surface markers do change and they alert the immune system to basically get rid of this cell. All right, so an antigen is a big molecule, and on a big molecule, you have little parts to it, right? So your immune system, the receptors of the immune system, doesn't recognize the entire antigen. It recognizes little pieces of it. So little pieces of it are just called epitopes. And that's all you need to know here. It says the definition of an epitope is a specific region of an antigen that the immune system can actually bond with the receptor for. Okay. So they call them motifs here. Any given antigen may contain several motifs that are recognized by immune cells. All right, so this is our protein. So let's say that this gray area is a protein. It is, the entire thing is an antigen. We have all these epitopes, these specific areas of our antigen. And you can see that the antibody is present. So antibodies always have this Y shape to them. So we have a green antibody, we have a red antibody, and they're color coded so they can show you that antibodies are going to recognize just one specific epitope. So the red antibody only recognizes the red one, not the green or the yellow. The green antibody only recognizes the green one. And the yellow one only recognizes the yellow one. So they're very specific to the epitope type that they can bind to. So antigenicity is a word that refers to how strongly or how weakly this antigen can provoke an immune response. And generally speaking, the larger and more protein-based the molecule is, the stronger the immune response, or it's more antigenic, right? So if something's a weak antigen, that's going to be a small molecule, more carbohydrate based. And so it just doesn't alert the immune system. It doesn't cause tight binding to those antibody receptors of B cells. And so you're going to get a weaker response. So there is a scale, right? There's a scale between, like, if a foreign molecule is very antigenic, then it provokes a strong immune response, all the way to a foreign molecule being weakly antigenic and creating a weaker immune response. So now that we've talked about antigens, let's talk about an antibody. An antibody made by your B cells. In fact, they're made by an activated B cell called a plasma cell. These are also proteins. Well, antigens can be anything, but antibodies are always proteins. And the proteins that form an antibody are basically, bound together to form this Y shape. There's two regions that you want to know about. There's the variable region and the constant region. The variable region is an antigen binding region. So the FAB stands for the fragment of antigen binding, here, FAB. And the constant region is not, the C here is not for constant. The C is actually for fragment of crystallization. But this is the area that docks onto different cells. So what we want to remember is the constant region and the FC region are basically almost the same, right? The constant region contains the FC region, and this stem part of the antibody is what can either is bound to, I'm just going to simplify the antibody here, right? The stem part is bound in the plasma membrane of B cells. Okay, so it serves as a receptor, that same antibody. And if B cells secreted these antibodies into the bloodstream, it's the same antibody molecule, but just released as a free-floating protein in the bloodstream. Sometimes these antibodies, this constant region, and you'll see this in allergy, other cells that are around, like a mast cell, for example, or a basophil for allergy, will have docking sites. They'll have little binding docking sites for these antibodies. and it's the FC region that comes in, and it'll just stick like this to cells. Okay. So that's the constant region. The variable region has two parts, right? You can see there's this region here, and so they can bind to the antigens. Both of these variable regions are exactly the same. They bind to the same, let's just use the word epitope, right? They bind to the same epitope. And what's interesting about the variable region is that every single B cell that your body has inside of it, the millions and millions, they each have a slightly different variable region. So it can differ in shape just by a small amount all the way to a large amount. And so the ability to bind to different epitopes, right? You have basically all these different kinds of antibodies that are ready, and anything that comes into your body, there's some kind of, the variable region of some of those B cells will bind it tightly. And then that tight binding will cause the B cells to be stimulated. Okay. So this is all a genetic shuffling that causes these variable regions to be different. What you want to know about the antibodies is that we want to know the different kinds, right? So the classes of antibodies are listed here: IgG, IgM, IgA, IgD, and IgE. We want to know the classes. We want to know what they look like, right? The structure of them. So IgG is a Y, just one single Y. IgM is a pentamer. So there's 5 different antibodies that are linked together by a protein. IgA is a dimer. So there's 2 antibodies hooked together and attached together like this. And then IgD and IgE are both the same as IgG, which they're just like a single antibody. So we want to know the structure, and we also want to know the function. Okay. So we'll get to some of the functions here. We'll talk about them. I want to point out that IgD is a B cell receptor. So that antibody just remains on the surface of B cells. Your naive B cells, every B cell that you have in your body right now that has not encountered its antigen, they're called naive. And these serve as their receptors. So these are all IgD. However, when these B cells get stimulated, there's a class switch, and so even though it has IgD on the surface, either IgM or IgG will be secreted. Let me point out that these IgEs. Think about IgE, allergy, very long E at the end. These guys are the ones I was talking about, that their stem, their constant region, their FC region is the one that binds to your basophils and mast cells that you can see here. And they are going to be very active against your allergens. For IgA, you want to remember that it's secreted. So neutralization and trapping pathogens in mucus. All right, so this IgA is found in your bodily fluids. It's going to be found in breast milk. It's going to be found in mucus. It's going to be found in saliva. So it's going to be found in places that are secreted out of the body. IgM is going to do a bunch of stuff. We can see here: neutralization, agglutination, complement activation. But you notice how it's so large. It can't leave the bloodstream. So IgM is going to remain in the bloodstream, which means it cannot cross through the blood vessels. It cannot go into the tissues to help fight infections in the tissues. So it's basically this really big, heavy antibody structure, but it's really, really good at finding the wrong blood type. So if you're getting a transfusion and the wrong blood type comes in, IgM is going to be the one that agglutinates it. And then for IgG, it does everything. So we'll talk about IgG, each of those actions. So one action of antibodies is that they neutralize. What that means is that these antibodies, right, this is an antibody, it will recognize the spikes, let's say, of a virus. And remember how viruses, they have to be able to fit into a receptor on your cells, right? So this virus, it has a little spike. These viral spikes actually have to fit a receptor into our cells, or some kind of protein, and has to like attach to it. And then our cells will bring that virus in. So if you were to put antibodies all over these spikes, right, bind to those spikes can never interact with the host. So that bringing in the virus never happens. So you've neutralized the virus. You can also see that neutralization also refers to toxins. So toxins, the active site, usually the toxins have like an active part that binds to the cells and causes issues. When antibodies bind to them, it blocks that part from being active. So it basically negates the action of that toxin. And then you can see antibodies can also prevent or could bind to like parts of the bacteria, like the flagella. And sometimes the flagellum actually causes that adherence or docking onto your body. So without that, it can't stay in your body to attach to the target cells. And maybe something like the mucus can push it out of your body. So that's neutralization. Opsonization is enhancing phagocytosis. So this is a long word. It just means enhancing phagocytosis. And you can see that here's our pathogen. Our antibodies have bound to some epitope, right? The variable regions have bound, the tips of the Y. But that FC region, hey, it's docked onto a receptor on the phagocyte, on this macrophage, for example. So when you're able to coat a pathogen with antibodies, and those antibodies have a specific protein for them on the phagocyte, this is going to help that phagocyte attach to the pathogen much better, right? Because you have these antibodies with the perfect FC region that fits your receptor. And it can, basically just lock onto the pathogen a lot more firmly, right? And to enhance the phagocytotic reaction. So that is really great for, your non-specific immunity to get rid of, to basically through phagocytosis, right? Macrophages, dendritic cells, and neutrophils are the big three phagocytes that use this receptor. Agglutination, so this is where we see the IgM, right? This is IgM. And this is where the antibodies are going to clump or bring together a different kind, or like the same antigen all together to make a really big complex. To agglutinate is basically to make a big clump, right? And so what this antibody can do is really grab onto many, many, many epitopes of a bacteria. It's gonna be bacteria, it could be red blood cell, but it can really form a huge aggregate of this certain pathogen. And the bigger something is, the more likely your immune system's gonna see it, right? The more likely phagocytes or something, even, specific immunity might be able to detect it and start to get rid of it, start to phagocytose the agglutinate. So that is another job of antibodies. And we have antibody-mediated cellular toxicity. So this is where antibodies help out T cells. Remember, antibodies are made by B cells. B cells, as we can see, these antibodies helped. Let's just go back, right? What are we helping? So we're helping, the antibodies are helping things dissolved in your fluids, right? Things are just like free floating, they can get neutralized. Antibodies made by the B cells are helping out your non-specific immune system. So your specific immune system is helping out your non-specific immune system. This is also going to help out your non-specific immune system by making something very large and obvious. And here we have an example of antibodies helping out your T cells. So when a pathogen has antibody bound to it, you can see that we have an FC receptor again for that FC region. And once you have binding, this T cell knows, hey, the antibodies are on this foreign invader. We should kill this cell. So the antibody's bound with the cytotoxic T cells or a natural killer cell, either one. These guys can say, basically the antibody is a flagging to the T cells or natural killer cells to kill it, to kill the foreign invader because it has those antibodies on it. All right, let's talk about a little bit different right now. There's two molecules we need to understand, MHC1 and MHC2. MHC stands for major histocompatibility complex. We have two in our body. This is the one that is going to be your self molecule. So we talk about how does your immune system know your cells from other people's cells? This is the one. So the MHC class 1 and class 2, those are going to be only your own. You have a very unique individual MHC. Unless you have an identical twin, you will not have the same MHC as anybody else in the world, which makes organ transplanting very difficult, right? Because MHC class 1 are found on all nucleated body cells. So what does that mean? If it's nucleated, anything with a nucleus, but there's one kind of cell without a nucleus in our body, and those are blood cells. That is why we can donate blood to one another, because blood cells do not have, and I'm talking about red blood cells, red blood cells don't have the MHC type 1. They do not have a nucleus. So you can, as long as the A and B antigens match, you can, donate blood. MHC class 2 is a little bit more specialized. MHC class 2 are found on macrophages, dendritic cells, and B cells. So look, these guys also have MHC class 1. But MHC class 2 is kind of a specialized receptor that is found on these specific phagocytes, right? Macrophages are phagocytic, dendritic cells are phagocytic, they're under the skin and mucous membranes, and B cells are also phagocytic. B cells are going to be found in the bloodstream and also in your lymphatic system. So when they recognize, so these guys are going to take up your antigen, process it, and then stick it on an MHC class 2. And the MHC class 2 is going to trigger your T cells. Okay, so this is basically a very important receptor for T cells to recognize. And MHC class 1 is for everything, right? MHC class 1, if it's different, then T cells natural killer cells will also figure that out and kill those cells. So you can see they're slightly different. There's no need to memorize the exact structure. Your book does make reference to the cleft being a little bit different where it binds to antigen, but you just need to remember that MHC class 1 is on all nucleated body cells, and then this one is on specific, what we call professional antigen presenting cells. All right, so this is that presentation I was talking about. What does it mean to present an antigen? So this guy right here, this is going to be one of those professional phagocytes I just talked about. Okay, so B cells, macrophages, dendritic cells. Those are the three that are going to have your MHC class 2. Okay, so what these guys will do is that they will engulf, right, endocytose, phagocytose your pathogen. And then once it's in this phagosome, right, then we're going to lyse it with our lysosomes, fusion with the lysosome. And then once you have destruction and digestion, the cell will choose different epitopes to attach to the MHC class 2, right? So you see how the MHC class 2 and class 1, they have an antigen binding site. That's where that little snippet of what we just caught gets put. And then the MHC class 2 goes up to the surface, and the immunodominant epitopes, which just simply means that the epitopes that this cell has chosen is usually protein-based. And so the protein-based is a better antigen, right, than something that's carbohydrate-based. and they put it on the cells. So what this does, what this is doing right here, right, is saying, alerting your T cells. It's alerting your T cells, specifically CD4 positive. Okay, adding a little bit of more detail here. CD4 positive T cells, there's two kinds of T cells, CD4 positive and CD8 positive. We'll talk about that as well in the future. This is a T helper cell, and T helper cells with a little h, T helper cells are really important in alerting all different kinds of parts of the immune system. So it activates the nonspecific immune defense, it activates other T cells, and it also activates B cells. So when a T helper cell recognizes this complex with the MHC class II, this cell is basically saying, hey, T cell, this is what we have. This is what we have in the environment. We need to get all parts of the immune system working. The T helper cell will recognize this. And then what the T helper cell does is basically just shoot out a bunch of cytokines, right? It's the alert. And those cytokines will go and activate the other parts of the immune system. So the MHC class II is really for these T helper cells to recognize, right? But that's what it is. So we're chopping up, processing the pathogen, making sure that we're choosing proteinaceous antigens or epitopes, putting them onto an MHC class II, putting it on the surface of this cell so that we can show our T helper cells what we've caught and to alert the immune system. This is a T cell, and it's just kind of cool to see all these protrusions of the membrane. It's basically like little finger-like projections, and that's to increase the surface area so they can, be in touch with the outside world a little bit more. Here's a picture of the lymphatic system. So this is where most of our T cells and B cells are, right? Our lymphocytes are largely located in the lymphatic system. So our lymphatic system is a series of vessels. You can see all these lines. They're all the lymphatic vessels. They basically take the fluid from your tissues, and they make sure that the fluid that comes into those vessels pass through lymph nodes. So there's lots of lymph nodes in your armpit. There's lymph nodes throughout your body. But what lymph nodes do, let me show you a lymph node, is lymph nodes will have it's like a densely packed little bean of B cells and T cells, dendritic cells, macrophages. So it's all these white blood cells located inside. And the lymph, you can see moving in. This is the fluid that came from your tissues, right? And notice that we have valves. The valves prevent backflow, so we don't have a backflow of lymph. It pushes that fluid through this. So imagine coming in, right? You have some water, this fluid coming in, and then you have this really dense area of cells, and you only have one exit, or I guess there's two exits, right? So not many exits, but we have 4 entrances. So we're going to get a huge backlog of fluid, and it's going to slow down as it moves through a lymph node. And so that slowdown really helps those B cells and T cells. If there's any antigen present in that lymph, then hopefully the B cell, T cell, dendritic cells, macrophages, these are our antigen presenting cells, they're going to be able to find it and get activated. So back to this picture. Our tonsils, we have lots of lymph nodes here in their neck, which is why your doctor will feel your neck if your throat is sore. One lymphatic organ we want to pay a little bit of special attention to is the thymus. And the thymus does shrink as you age. So a newborn has a really large thymus, and adults, it shrinks. And sometimes in older adults, it just disappears completely. But the T in thymus is important because that's why T cells are called T cells. The T stands for thymus. And what happens is what the thymus does is continues to produce T cells into adulthood. And they used to say, and I also learned that like when you're 40 or 45, you basically don't have a functional thymus, but they're finding that you do. It may help, even though the thymus's gland functions decline over time, it may help adults fight viral infections. And T cells are correlated with cancer. So T cells are able to, basically T cells are able to determine whether your cells are self, if they're good, you know, if they're self cells that are healthy, they leave them alone. This is supposed to say self, by the way. And if they're non-self, right? If your cells look like they are starting to change, those MHC class 1s don't look right, or they're virally infected, then the T cells will basically kill non-self. So down here, you see that there's a study people who had their thymus removed had higher rates of cancer, autoimmune disease, and death, because the T cells are the ones that are surveying your body for precancerous cells. Any cells that are starting to become cancerous will change how it looks on the outside, and it will, and that's the job of the T cell to fight that. So the job of the thymus, again, is to mature your lymphocytes and learn. What that means is it learns what's self and versus non-self. And so it's available to produce these T cells over the lifetime, but definitely goes down as you age. All right, we'll stop that.